Hello and welcome everybody for this session. So this week's paper is going to be this examination from 2016, and this paper is an AS paper. So as always, I'm going to try and go through this as quickly as possible, concentrating on maybe some of the less obvious bits, because hopefully you can all check some of the easier answers from the mark scheme. So let's get straight into it. So the first question. Figure 1.1 shows 4 animal cells in different stages of the mitotic cell cycle. Using the number given to each cell in figure 1.1, arrange the stages as they occur in the meiotic cell cycle. Okay, so having a look at it, in cell 2, you can see that the chromosomes are uncondensed, or starting to condense rather, and the nuclear envelope is breaking down. So this is going to be the first step. And then in cell 3, you can see that the chromosomes have indeed condensed, and they have lined up on the equator. And you can also see the spindle forming and attaching to the centromeres. So this will be number 2. I mean, cell 3 is going to be the second step. And then in this one, you can see that the sister chromatids have separated, and so this one would be anaphase, and they have migrated to each of the poles of the cell with the help of the spindle fibers. So this one is the third one. And then finally here, you can see that the chromosomes are starting to decondense again, the nuclear envelope forming and cytokinesis taking place. So this will be the last one. And then the cycle would, after some growth, the cycle would start again. Okay, next one. Also, this one is number 1, 2, 3, 4, 3, and 4. Okay. State what's occurring at A in cell 2. So this one is, as I've mentioned just a minute ago, this one is the nuclear envelope breaking down or disassembling so that the chromosomes can be separated. Okay, and then label B is pointing to a region of the chromatid that contains repetitive nucleotide sequences. State the name given to this region. So this is necessary. So it's called a telomere, and it's found at the end of each chromosome, and it's to protect the coding regions of the chromosomes due to a fragmentation that occurs at every single copying of the chromosome sequence. Okay, the centriole labeled in figure 1.1 is composed of microtubules. Suggest why a student would not be able to see a microtubule using a good quality light microscope. So outer diameter, because it's a hollow tube, of a microtubule is around 25 nanometers, and the highest resolving power of a light microscope is around 200 nanometers, so you wouldn't be able to resolve the microtubules because the resolving power is limited by the wavelength of light. Okay, second one, outline the role of microtubules in mitosis. So mitosis forms part of the spindle fibers, and they attach to the centromeres on the chromosomes, and they help the movement of the sister chromatids to opposite poles, and therefore helping in the process of forming two daughter cells in mitosis. Okay, microtubules are composed of a globular protein known as tubulin. Explain how the terms monomer and macromolecule can be applied to tubulin. So a microtubule is actually composed of 2 monomers, so it's alpha and beta, and they form dimers. And what happens is, as they polymerize, they kind of form this sort of spiral, like so. And in this process, you end up with this hollow tube. So the monomer is this individual building block of the tubulin, of the microtubule. So it's a protein which is composed of polypeptides, and when polymerization takes place, you get this macromolecule by joining together the monomers via peptide bonds. Okay, second question. Figure 2.1 shows the central region of a transverse section of the root of a dicot plant. Okay, so draw an arrow on figure 2.1 beginning at point W and ending in a xylem vessel element to show the direction of water movement in this region of the root. So in the root, this sort of cross shape is going to be your xylem, and water moves from these parenchyma cells into the xylem, so it would be in this direction, so into any of these vessel elements. And then describe the pathway that water takes from point W to reach a xylem vessel element, and explain why water moves in this direction. So from the soil to get to the xylem, there's actually two parallel pathways, the apoplastic and the symplastic pathway. And the apoplastic pathway is sort of faster because it moves outside of the cytoplasmic region, so it moves through cell walls. However, it's limited in movement once because parent strip is reached, so it has to diffuse into the endodermis via the symplastic route, which is to say water moving from cell to cell through plasmodesmata inside the cell membrane. So in this case, we would describe the symplastic flow through the cytoplasm via the plasmodesmata. So in the context of the parenchyma cell to the endodermal cells, it would move from a higher water potential than the adjacent cells. So overall diffusion takes place across membranes from a higher water potential to lower water potential. Similarly, how it diffuses from a higher water potential in the soil into the xylem. Okay. The parenchyma cells in figure 2.1 are packed with starch granules, which are used as energy stores for the plant. The starch granules are not clearly visible in the section shown. Name the solution that can be used to make the starch granules more visible to confirm that the granules are composed of starch. So this one is the iodine that is found in potassium iodide would stain anything that contains starch into sort of a blue-black color. And then describe how the structure of starch makes it suitable for its function. So there's two different macromolecules that make up starch. One of them forms an alpha helix, and that one is amylose. And then there's also a myelopectin, which occasionally, when two glucose molecules form a different kind of bond, because of steric hindrance, it forms sort of a branch, so it looks more like that. And then, so the reason why the structure of starch is useful is, first of all, when you've condensed your glucose into a macromolecule, it's no longer soluble, so it's not osmotically active. So it's a way to sort of lower the concentration of glucose in the cell without having to transport it away from the site. So, and also it's very compact for storage. And amylopectin, because of this branch nature, has many free ends, so it can supply glucose very quickly, because you can have a hydrolysis reaction here, and here, all at once, whereas in amylose you could have a hydrolysis reaction at either end of this macromolecule. So yeah, it's mostly very useful that the glucose needed for respiration it contains glucose, and it's present right there in the cell. Okay, question 3. Figure 3.1 is a vertical section of the heart. The regions concerned with initiating and conducting impulses are shown. Name chamber B and blood vessel Q. So, firstly, P would be the right atrium. So, and then Q, the biggest and strongest vein in the body, actually an artery, sorry, is called the aorta, which pumps oxygen-rich blood all over the body. Describe how the sinoatrial node and Purkinje tissue shown in figure 3.1 are involved in the initiation and coordination of the contraction of the heart. So first, let's talk about the sinoatrial node. So this is sort of a region, a patch of cells that's the natural pacemaker in the heart. And what happens is that from nerve impulses, from, let's say, the vagus nerve is one example, it sets a rate of heartbeat, and the action potential starts from these patches of cells, and then it spreads in a controlled manner throughout the heart, and that controls the rhythmic contraction of the muscle. So firstly, it brings about the contraction of the atria, and the atrioventricular node is, once the impulse reaches the atrioventricular nodes, there's a delay in sending the impulse around to the rest of the heart, so down into the ventricles, and this is to make sure that the atria have had a chance to fully contract before the ventricles do, so that the flow of blood is as efficient as possible. And I think that's it. So the movement of the blood would be like so. But if you look at the valves, you can see that, so these are all to prevent the blood from flowing backwards. So you can see that they would move. So this part, the atria would contract first, and then the ventricles would, and it can send the blood either around the body, or in the case of the pulmonary artery, into the lungs to enrich the blood with oxygen. Okay, and then Purkinje tissue. So the Purkinje tissue, we've already mentioned this, but this is what's responsible for conducting the impulses to the ventricles, to the base of the heart, and then it conducts the waves of excitations through the ventricle walls causing the ventricular contraction, or in other words, the term to use is systole. Okay. The heart forms a closed double circulation, a part of a closed double circulation in mammals. Explain what is meant by closed double circulation. So closed just means that the blood is contained in the blood vessels, the arteries and the veins and the capillaries. And then double means that blood travels through the heart twice during one complete circuit. So there's the pulmonary circulation and the systemic circulation systems. So in the right side of the heart, the oxygen-poor blood is pumped into the lungs, and then at the alveoli, gas exchange takes place, and blood returns to the to the left side of the heart via the pulmonary vein, and then that oxygen-enriched blood then gets pumped around the body through the aorta, and you can see that that's why this side of the heart is slightly it's not. This part of the heart is slightly bigger, is because a stronger contraction is needed to pump the blood all around the body. Okay. The blood in the pulmonary vein has traveled through the capillary network in the lungs, where gas exchange has taken place. Describe the process of oxygen transfer between the alveolus and the blood in the surrounding capillary. So if this is the alveolus, and then let me use a different color for the blood vessel, going around it like this. So essentially what happens is that if the flow of the blood is this way, so most of the blood is saturated with carbon dioxide here, and then what happens is that through diffusion through the alveolar wall and the capillary wall, so the endothelium, gas exchange can take place. So this is all to do with concentrations. So if the concentration of carbon dioxide is high here and lower here, it gets pumped out. So that's CO2, sorry if that's a bit small. And then there's a high concentration of oxygen here and low concentration of oxygen there. So then oxygen can diffuse into through the cell walls, and then oxygen can be taken up by heme in the hemoglobin and then taken back to the heart and pumped around the body. When a person starts to smoke cigarettes, one immediate effect is a decrease in the oxygen that's taken up into the lungs. Explain why this is so. So this is to do with the nature of hemoglobin. So when you smoke a cigarette, sometimes it doesn't combust fully and properly to release carbon dioxide, so some carbon monoxide is made, and the hemoglobin can interact with the carbon monoxide much more strongly and easily than with oxygen. So essentially it competes with oxygen for the binding site on the hemoglobin, and so overall there is a lower uptake of oxygen in the lungs. So it sounds horrible. Phosphate ions have a number of uses in organisms. These include involvement in cell signaling responses, involvement in energy transfer processes. They're a component of phospholipids and nucleotides. Phosphatase enzymes remove phosphate groups from organic compounds, while kinase enzymes add phosphate groups. Read the following passage. Some hormones circulating in the blood are able to trigger transcription within a cell, even though they're unable to enter the cell. Phosphatases and kinases then take part in cell activities that eventually result in genes switching on and transcription beginning. Suggests why the hormones referred to in this passage are unable to enter the cell. So these hormones are they can combine with cell surface receptors on the target cells. So there's a variety of reasons why they can't diffuse into the cell. One of the reasons is they don't necessarily need to, because the cascade that they trigger inside the cell is already sufficient and, in fact, probably more controlled. So there are some steroids specifically that act directly on gene transcription, but the majority will just bind to the cell surface receptors where they trigger a cascade of synthesizing firstly a second messenger and then activating phosphatases and kinases to carry out the cellular processes that will eventually result in triggering transcription. So they're normally too large. They could be too large to cross the membrane. They are almost always hydrophilic, so they can't pass through easily the hydrophobic membrane, and they probably don't have any transport proteins to help their diffusion across the membrane. Okay, and then use the information in the passage to outline the process of cell signaling. So we've already kind of gone through this, but once again, the chemicals, the hormones are released from an organ, and then they travel to the target cells where they combine with the cell surface receptors that trigger the action of kinases downstream, and eventually lead to the specific response, which in this case is transcription, not activation. The activity of a phosphatase enzyme was measured at different values of pH by using 9 different buffer solutions. The temperature was kept constant at 30 degrees Celsius. So here are the results. Disrupted. So either the active site is changed so that the enzyme can no longer bind to its target as efficiently, or the tertiary structure can be fully disrupted and a denaturation of the protein can also occur. Okay. DNA can be produced commercially for use in genetic engineering. Sometimes dephosphorylated DNA is required. This involves removal of the terminal phosphate groups in using immobilized phosphatases. State one way of immobilizing an enzyme. So one easy one to remember is to have your enzymes in sodium alginate beads. So they are encapsulated in this bead where they can be still active at the interface of the bead, but they're not released into the surrounding area. Apart from a cost benefit, suggests one advantage of using immobilized phosphatase to produce dephosphorylated DNA. So we can mention a number of things here. So first of all, so that the enzymes can be reused. Secondly, the enzyme is really easily recovered and there's no need for purification of the product. So it can be recovered, but also it helps with the purity of your product, so it doesn't contaminate it. And therefore, the enzyme has a longer shelf life as well. And sometimes it's even more stable to any other influences, such as the change in temperature or pH. Okay, so this is a nucleotide, and the question is asking us to compare the structure of an ATP molecule with the DNA nucleotide. So an ATP molecule would look something like, I'm just going to draw it here, ATP. So we've got our ribose sugar, and then we've got our adenine, and then triphosphate. So they do look quite similar. So some of the similarities, as I've just mentioned, are the fact that they both have pentose sugar, and they both have a nitrogenous base attached to that sugar. And furthermore, finally, they both have phosphate. So their overall structure is very similar. However, some of the differences are that in ATP, the sugar is ribose, not deoxyribose like it is in DNA. And then obviously from the A in ATP, you know that the base is adenine. So in this case, on the DNA is guanine. So they both still have purines, but they're different bases. And then finally, ATP has a triphosphate group here, whereas DNA only has one. Okay. Question 5. Complete table 5.1 to show which of the following statements apply to each of the infectious diseases shown. So unfortunately, this one is something that you need to know. So which one is caused by a virus and so on. So measles is a viral disease, and so is smallpox. However, malaria isn't. And then there we go. So the second one is asking which one of these is caused by Plasmodium. So we know that that's a parasite, and it causes malaria. Eradicated by vaccination, so I think it's the only one still. I know mumps was close to being eradicated, I think, but there's been some flare-ups due to the anti-vaccine movements, especially in the US. So smallpox is the only illness that is to be eradicated by humans. So unfortunately, neither of those are eradicated at this point. And then none of these are transmitted by contaminated water. Okay. Explain why antibiotics are not prescribed for viral diseases. So hopefully this one is an easy one to finish with. So the idea is that they are completely different organisms, if you are to call a virus an organism in the first place, which is questionable. But viruses have no sites, no targets for the antibiotics where they can bind and sort of disrupt their spread. So normally, secondly, viruses are normally located inside infected cells. So most of the time, even if the antibiotic could have an effect on the virus, they probably wouldn't be very effective because the viruses are normally located inside the cells. So yeah, anyone who prescribes an antibiotic for you when you have just a cold is not being careful enough. Okay, so that was the last question. So, oh no, it wasn't, sorry. My bad. One more. Question 6. Figure 6.1 shows the origin and development of a B lymphocyte and its subsequent role in an immune response following an infection by a pathogen. So as we can see, from a stem cell, the B lymphocytes mature, and then once they're exposed to an antigen, they can take up 2 fates. So they can develop into a memory cell or a plasma cell, which then produces the antibody against the antigen that they were exposed to. B lymphocyte activation can occur as a result of direct contact with a specific antigen on the invading pathogen. State 2 other ways for the B lymphocyte to become exposed to the antigen. So we've just been talking about vaccines, so that's one of them. And then a second way could be from an antigen presenting cell. So an immune cell that has been infected by the pathogen, and then the phagocytosed pathogen, the pathogen will be broken down, and then the antigen will be presented on the infected cell, on the surface of the infected cell, to which the B lymphocytes can bind and get activated. Okay. The function of the plasma cell is to produce and secrete a specific antibody. Complete figure 6.2 to summarize the sequence of events occurring in the plasma cell. So the nucleotide sequence on the DNA template strand is copied and mRNA is synthesized. So this one is transcription, which occurs in the nucleus. And then the polypeptide is synthesized, so this one is translation, occurring at the ribosome. And then the antibody is produced by further processing. So this one, depending on what the modification is, can either take place in the rough endoplasmic reticulum or the Golgi body. Okay. Antibodies are globular proteins with quaternary structure. Suggest why antibodies need to be globular proteins. So this is so that they're soluble in the blood or in any other tissue fluid so that they can get to their target. And the quaternary structure and the tertiary structure, in fact, allows for the formation of the variable site as well. So if it was a fibrous protein, it would be very difficult to transport it to the site of infection. So that's why they need to be a globular protein. With reference to antibody structure, explain what's meant by quaternary protein structure. So quaternary protein structure simply means that it's made up from more than one polypeptide. And in the case of antibodies, they have 2 heavy and 2 light chains, and I think 4 different polypeptide chains, I think, and they have a variable and a constant region. And then finally, this time, monoclonal antibody can be produced commercially for use in the diagnosis of infectious disease. Name the type of cell used to produce monoclonal antibody. So this one is called a hybridoma. and it's essentially a B cell that's been fused with myeloma cells, so that they can take the beneficial part of a cancer cell, which is that the cells are immortal and they can grow in tissue culture. So if those two cells are fused, then the monoclonal antibody can be produced at a massive scale, and then the tissues can be cultured. Sorry, the cells can be cultured almost indefinitely due to the property of the cancer cell. Okay, so that's the last question. Hopefully that was useful, and thanks for tuning in again. I'll see you next time.